Hello, welcome to my scenario. Have you ever wondered why there are billions of people in the world, but each one looks different? Well, that is because each person has a unique set of genes. To start off, genes are the most basic building blocks of humans. They are made up of DNA, which include 4 proteins: adenine, thymine, guanine, and cytosine, or commonly referred to as A, T, G, and C, and these, with a phosphate base, make up DNA. A bundle of genes that make up a certain trait of someone is called an allele, and a bunch of alleles wound tightly around each other form make a chromosome, which looks like an X. Now there are 46 chromosomes, or 23 pairs, in every single nucleus of a cell, with trillions of cells in your body. With all those strands of genes, less than 1% of them differ between every human. And all these facts would be lost without the help of Gregor Mendel, who recognized the first idea of genes and heredity. His work included breeding pea plants and seeing the similar traits being passed down as they were reproduced. With this basic information, everything we now know of genetics is based off Mendel's discovery. With the basics done, let's talk about heredity. You are given 2 sets of genes for every part or trait of your body, one set from your mom and one from your dad. Now in those genes, to determine which trait you end up having, we have to look at dominant and recessive genes. For example, having brown eyes is a dominant gene, and this means if your mom has brown eyes and your dad has blue, you are more likely to have brown eyes opposed to blue or green. This is because any dominant gene produces enough related proteins to have a signature trait, and a recessive gene lacks these proteins. Therefore, the dominant trait takes over and is physically shown. With brown eyes, the proteins for that are coded to make more melatonin, which give it that dark color. But with 2 recessive alleles, such as having blue eyes, you will also have blue eyes because there is not enough melatonin coded to produce to make it that dark color. This reasoning was based off Gregor Mendel when breeding a tall pea plant with a small pea plant. It most of the time turned out to be tall. He figured out dominant and recessive traits, with the tallness of the pea plants being dominant. But of course, the human body is not always that simple, and your grandparents can play a role in your traits. A visual representation to show this is the Punnett square. Going horizontally is the father's genes, and vertically is the mother's genes. The capital T represents the gene for tall, and the lowercase t is the gene for short. Both plants have the genes capital T, lowercase t. The lowercase t means that each plant is a carrier for short from their parents, but they both physically show the tall trait because it is dominant. So the offspring can take any one of these alleles as their DNA. When reproducing, there is a chance that the pea plant can take the bottom right corner and appear short, but since it is a recessive gene, there is only a 25% chance of that happening. Just knowing the dominant and recessive trait cannot always determine the outcome of a child's traits, but they can be very helpful in giving you the odds. And this is the same with brown or blue eyes, with the capital B being brown eyes and the lowercase b being blue eyes. And if the father was not a carrier for the blue-eyed gene, then the child would have a 100% chance of having brown eyes, but has a 50% chance of being a carrier. And if the mother had blue eyes and the father was a carrier for the blue eyes, then the child would have a 50% chance of having blue eyes. And if the father had blue eyes, then there's a 100% chance that the child would have blue eyes. Not all traits are dominant and recessive, though. Some traits are codominant. This means that you have a blend of traits that your parents have. An example of this is flowers. If you have a flower with a red color and a flower with a white color, their offspring will have a pink color. It can be confusing, but what is more confusing is genes with mutations. Learning about genes has helped us to learn more about mutations and conditions that people have. A mutation is a coding error that your gene gets when being copied from your parents into you, and they can sometimes lead to physical abnormalities. Genes with mutations are surprisingly very common, with people averaging 5 to 10 genes with mutations in every cell. But as I said, since we have 2 copies of every gene, the mutation is usually covered up by the normal copy, and this is called an autosomal recessive. Now, with mutations in the chromosome, something neat is that boys are more likely to have a mutational condition than girls. I'll explain. Girls have 2 X chromosomes, one from their mother and one from the father. Boys have an X chromosome from their mother, but what makes them physically like a male is the Y chromosome from the father. With that in mind, if something is wrong or mutated with a boy's X chromosome, that mutation is shown, whereas a girl with a mutated X chromosome has another X chromosome as a backup. So, a condition in the X chromosome like color blindness is more likely to occur in boys because they only have one X chromosome. Color blindness can still occur in girls though, they just need a mutation on both chromosomes from both parents. Things can also happen where you inherit too many or too few chromosomes, with a popular example of this being Down syndrome, and that means the person has an extra chromosome. All this information, we are still learning more and more about genes and their role in the human body. For instance, gene mapping, which is getting a sample of a person's DNA and seeing the order of proteins to determine different traits or diseases they could get. One example of this is a TED Talk, where a man named Dr. Greg Venter's entire genome sequence, meaning every single gene, was figured out and mapped in books. For just one person, there is 262,000 pages of the four letters A, G, C, and T over and over again, and the letters actually mean something. For example, a line of many pages of many books read AG, AAG, AAT, ATA, and this gives him his eye color. With all this advanced technology, we are able to figure out more about life than ever before, and the advancements could really make a difference. One example of this is gene mapping in chromosomes and using single nucleotide polymorphisms, or also known as SNPs. SNPs are single base pair polymorphic regions, or to make it simpler, SNPs detect the less than 1% difference between humans' genes. This can be especially helpful when trying to find disease-associated genes. By now, a question you might have is, why does this all matter? Well, mapping genes can help to treat or prevent diseases or mutations people might have. An example of this you might know is the famous actress Angelina Jolie. She had a high risk of ovarian cancer in her genes, and she knew this from her family's history. While those cancers usually are not associated with a genetic cause, her family's genes were part of the 5% of breast cancer patients that have genetics to blame, and 15% for ovarian cancer. Jolie carries the BRCA1 gene, which is a gene fault in her family line that makes them likely for these cancers. With this gene, she had a 50% chance of getting ovarian cancer and an 87% risk of getting breast cancer. Jolie then underwent the procedures of a double mastectomy and removing her ovarian and fallopian tubes to greatly reduce the risk of getting ovarian and breast cancer. The risk of her getting breast cancer dropped from 87% to under 5% after the surgery, which is a huge success. Without scientists learning how to gene map, Angelina Jolie and many others in similar cases to her would not be able to fix the odds that are stacked against them. Another idea that intrigues and sometimes instigates arguments is designer babies. With the ability to map out genes and see which genes give you which traits and having technology that can possibly alter those genes, parents could possibly change traits to what they want it to be and design their baby's features, designer babies. There are many different views and sides of the subject, and some people believe we should not be able to alter genes at all. Some believe changing any gene and designing a baby's traits is okay to do, and some believe it is okay to alter genes to prevent the baby from having a serious condition or disease. And of course, there can be many more opinions. One way to make sure a baby does not get a disease is using a method called PGT. This involves fertilizing the egg and seeing the DNA in the embryos to see which will carry a disease, selecting an embryo without it and putting it into the mother. People think this method could be taken too far and be used to get a superior embryo that has good traits like intelligence. But according to a November of 2019 study, the technology is not there yet and is too unreliable to see if picking traits like tall and intelligent would actually work. With new technology comes new power that we did not have before, and even though actually picking a baby's desired trait is not here or substantial yet, it is coming in the very near future. The simple act of a pea experiment by Gregor Mendel has led to making serious diseases preventable and possibly choosing a child's traits before they are born. I hope you learned something, and thank you for watching.